Free nucleotides. Nucleotides are the building blocks of the DNA strand. They have a sugar, phosphate, and a base. Topoisomerase is an enzyme whose function is to undo the double helix structure of the original strand of DNA, turning it into a zipper-like model. This is done before the split done by helicase. Helicase is an enzyme that is essential during DNA replication because it separates double-stranded DNA into single strands, allowing each strand to be copied semi-conservatively. Single-stranded binding proteins wrap single-stranded DNA to protect it from degradation and to make sure that it stays straight for the replication process. The replication fork. The split in the original DNA strand done by helicase, which leads to the formation of the lagging and leading strands. During DNA replication, one original molecule of DNA is split into its two strands, which in turn create two new molecules of DNA from the original one, each one having one strand of the original DNA molecule's DNA. This is called semiconservative replication. DNA replication occurs in an anti-parallel fashion because this configuration allows the DNA strands to form complementary pairs of their nucleotides using hydrogen bonds. The formation of the leading strand begins with one RNA primer, put there by the primase, after which the DNA polymerase III takes the template from that primer and begins adding on more DNA complementary of the original strand. For the 10 example base pairs, it could go AT, CG, CG, GC, GC, TA, GC, AT, GC, CG, AT. Unlike the leading strand, the lagging strand is created by many fragments called Okazaki fragments, each of which have their own RNA primer and are extended towards each other by the DNA polymerase III, essentially backwards from the replication fork. Ligase is an enzyme which connects the individual Okazaki fragments on the lagging strand of DNA by forming covalent bonds between them. DNA polymerase I is yet another enzyme which removes RNA primers from the leading and lagging strands and replaces them with regular DNA.